I'm Alexander Büsser. I'm an associate director at Idorsia. And to my right, for all of you who are new here, you may not have seen the talks, we have Jonas on my right-hand side, who is an expert in imaging or extracting biomarkers from images, and he's affiliated with numerous universities in Lausanne, as I was told. Then to my left, we have Félix, who is a lead data scientist at Okin, who has recently focused on using AI for covariate adjustments. And then we have Marianna, an IBMer, so working in research and focused on using machine learning to better understand the functions of single cells.
Thanks, Alex. So for the topic of panel discussion today, we have considered to talk about causal modeling in applications of machine learning in pharma, as well as application of multimodal data analysis, again for machine learning in pharma. And so why we have chosen this topic, I start with multimodal data analysis. I mean, as we all already know here, that technology has given us the advantage of getting data with different modalities from different sensory inputs. But now at this point, the challenge is that how we could get this depth understanding of combining this data all together. And also going towards to finding personalized medicine for patient. Machine learning could help us to find out what is the best way that we could combine this data all together to be able to find optimally prognostic and predictive biomarkers. But of course, all of this comes with several challenges that here I start asking our panelists that in their specific field of working, what are some of the challenges, for example, that they are facing when they are combining the data, for example, with data representation? Like, for example, in single cell analysis, what are some of the challenges that you have faced and some of the solutions that you could suggest to our audience?
Yes. So I would say, like in the single cell field, we're starting now to get a lot of experimental methods that are multimodal. So someone mentioned before CiteSeq, for example, that you can get protein expression and also mRNA expression and so on. So these are very, very exciting data, of course. Now, for the first time, we can quantify in the same cells exactly how much of each of these biological entities we have, and we can really start addressing some very fundamental questions, right? But then there are a lot of issues, such as, for example, I would say the biggest challenge is the fact that many of these modalities have very different dimensions. So in the case of mRNA, for example, you have tens of thousands, but you might have from the same cell only 40 or 50 proteins. And those proteins are only surface proteins, right? So you don't really know a lot about what the cell is actually doing. Maybe you can phenotype it, but you don't know exactly what the cell is actually doing. So one thing would be, what is the best way to capture all this information while taking into consideration the fact that you have so disparate data representations in the first place, right? So of course the most simplistic approach would be to progressively squeeze the data until you reach the same dimensions, but maybe this is not the best approach, right? And then another issue could be also the fact that these modalities are also affected by noise in very different ways. So sequencing approaches usually have a lot of missing data, and there were talks about that as well. So let's not get into that, but I would say like they have their own different peculiarities. So how the data is pre-processed is also very, very important. But I would say like ultimately the field is just starting now to understand what is the best way to integrate this data, and also some challenges have started to, like last year in Europe, there was a big challenge on single cell data integration. If anyone is interested, you can check out also the winning teams and so on. And then the main questions are, how can we predict like one modality from the other, right? So if we know, for example, the cell state by means of mRNA expression, can we predict protein expression, right? But also more exciting things are, how can we, for example, represent the data in a low-dimensional space that is fusing together all types of information we might have, and by doing that, are we able to find subcategories of cells that are predictive of disease outcome? But I would say we are now starting to address these questions, but it's already very exciting.
Yeah, great. So maybe I move on to Félix to explain his experience about application and challenges in multimodal data analysis for disease modeling in clinical trial.
Yes, absolutely. So, yeah, multimodality. So if you think about what I presented on covariate adjustment, you really want to show an increase in performance in C-index. So to show that you really capture more information by combining modalities than if you simply look at say only the clinical data. So that's not always obvious, and obviously I've focused on the cases where it worked, but there are many cases where we find information on histological slides, but does not allow to go beyond what is already predictable from the clinical data. Marianna mentioned the possibility of doing multimodality by predicting one modality from another. That's a very interesting concept and very fruitful. And for instance, we've worked on at Okin, we've worked on predicting from the H&E slides, so the histopathology slides, predicting the RNA sequencing data, so predicting expression data from the slides. And this works very well for some genes. It doesn't work for every gene, but it allows even to spatialize simply from histology plus bulk RNA-seq. You get a prediction from how much you're going to get expression of that gene, but you also get in the model interpretability, you get some spatial information of what does the model use to predict that the increase in expression will come, and so yeah, I think.
Thank you very much. And so I move on to Jonas. Yeah, we already take advantage of your great talk about what are the challenges of combining data in a federated way, but are there also some more challenges on top of that, combining the same data, but now the multimodal data in like when we are combining in federated way?
Yeah, so let's first start. Let's leave the federated out for the moment because it's
Sure.
already complicated. So okay, so I'm gonna walk back a little bit from the fancy analysis, the scRNA, etc., which is like super great to talk a little bit about like what you can do in a clinic currently and what is done currently in the clinic. And there's one term that maybe helps, which I don't know if it's how specific it is. It's called the clinical radiological paradox, and that's very famous for MS. Basically, it says that you have a clinical score on one side, and you have some imaging findings on the other one, and there's no correlation. An example for this is multiple sclerosis. You have this scale called the EDSS, the Extended Disability Status Scale by Kurske, which goes from 0 to 10. And you have, let's say, your imaging biomarker. Like typically, you could count the number of lesions that you have. If you look at the correlation between these two, it's really poor. It's really, I mean, horribly poor. I'm talking about, I don't know, if you get like 0.15 Spearman correlation, it's a good day, you know? So this is basically saying that the image, the biomarker you're taking from your image has no relationship to the data. In Alzheimer's disease, you have other scores. You have the MMSE, you have the ADAS-Cog, you have a number of scores, the CDR, Clinical Dementia Rating. And then your imaging biomarker could be typically, as I was mentioning, this hippocampal volume. Again, and so this is a bit better. Like this one correlation is indeed like you have less of a paradox between the two. But in the two fields, of course, like medical doctors are very interested in the pathogenesis of the disease and in actually looking. The images are one thing, but maybe you can get some other data. So for Alzheimer's disease, there's the two biomarkers, which are like the classic biomarkers of the disease, are called amyloid beta 142 and tau, the tau protein, phosphorylated tau protein specifically. These are things that you can get from lumbar puncture, for example. That's where you get the best readings. So you stick a needle in the back of the patient, you extract some fluid, then you can get your readings for these levels of these proteins. Okay, so on Alzheimer's disease, you can do that. And then in MS, clinicians are very excited about NFL, the neurofilament light. So this is another thing. You can also get it from the blood if you want. So this is another of these markers of inflammation. So now based on this, you have data sets that are open. In Alzheimer's disease, you have the ADNI dataset, which is an amazing resource that has all of these data that I mentioned. The proteomics is a bit limited, but nevertheless, you have like 100 patients with all this data. And this has been going on for more than 10 years, and I've also worked on this kind of, can I improve my correlation between like the clinical score and the prognosis of this patient, so the diagnosis of this patient, and the imaging if I add the biomarker, so if I have this multimodal data, right, if I have these protein levels. And the answer is, my answer is a little bit. Some other people's answer are yes, some other people's answer are no. So it's really like if you do meta-analysis, I think it helps a little bit, but maybe we're just doing it wrong. Maybe my feeling is that the data set we have access to, like clinical data set I'm talking about, they're just too small for now. It also might be, it's the same in multiple sclerosis. So if you go to this ECTRIMS conference, the big like European Congress on multiple sclerosis, people are very excited about neurofilament light. The problem is that it's sensitive, it's not very specific. So you have elevated NFL in a bunch of patients that have very different EDSS scores, so these clinical scores. And so again, probably this is also like we've tested this kind of association with a few hundred patients, getting close to 500 something, and it's not super brilliant for now. So I really don't want to say that multimodal data does not work. I hate this because it should, right? It's multi-view. Like in machine learning, you want to do multi-view data if you have orthogonality or some complementarity, but in your modalities it should help. So I think currently, at least for the clinical thing. The clinical aspect, I think probably we're doing late fusion, whereas we should be doing much more early fusion, probably. But it's really a mixed bag. So I'm hopeful. It's also like I'm doing research on this. So I really want this to work, but it helps a little bit, let's say. That's my current conclusion.
Thank you.
Thank you so much. Maybe moving from the theoretical to maybe a more practical question. So as computer scientists and data scientists, we're of course eager to maximize entropy and are eager to get our hands on different modalities. But particularly in a clinical setting, this comes at a cost for patients in doing additional imaging procedures, or in clinical trials, right, at the higher burden of the patient to report more information which can have an effect on the continuity of those patients in clinical trials. Are there any practical recommendations you have with this regard to make this trade-off between precision and respecting the well-being of the patient?
So yeah, I think that's a great question indeed, because as you said, we want to maybe maximize the number of modalities, but so this is one point that is not mentioned very often. So radiology now is all about this crazy about AI. So everything is AI. You have a lot of startups and you have a lot of things, but the question that is not very often asked is it really cost effective? So this is, sorry to be kind of a downer here, but this is like something in the end, like if you want to, let's say, buy a license for software that does the analysis on the image and all the omics you have, somewhere you have to bill this to the insurance company. So the way to do that is to prove that it's cost effective with respect to the standard of care. So you need to do a health economics analysis, right? And this means that the delta that you have, let's say you're talking about, like, essentially we want sensitivity and specificity. That's what we want to optimize. But you want to show that the delta, let's say you have your imaging and then you have your scRNA and then you have your cool whatever, the best omics, let's say your gut microbiome or whatever. So what you want to show is that you improve the care for the patient when you combine both in a way that does not cost too much in a sense. So we have right now, just I can mention this, that we have like one study now on maybe not multimodal, but we have a study with Patrick Omumi, a radiologist, on actually evaluating these automated fracture detection software in the clinic, because the claim from the vendors is that, okay, if you give this to emergency room technicians, they will make a mistake like 20% of the time, and then if you don't detect a fracture, it can go bad. So then the health outcome, it's bad health outcome. You have missed days of work, so you can quantify this thing. And if you use our software, you will get 96% accuracy, and it's good. But now we actually tested, and the ER physicians, at least in Switzerland, they're well trained. They're pretty good. So the delta to overcome this, it's not obvious that it's going to work. So this is work with also the team of Joachim Marti and the people in Unisanté. You have people that do here in Lausanne that do a lot of health economics exactly for this kind of things. That's a really interesting way to think about the problem. And then another thing, a project that was just approved now with Naghmeh Violi and Clarisse Dromain in CHUV is about like liver cancer. And then we want to see whether it actually makes sense to make this extra imaging analysis. Can we actually save the quality of life? So this is again a health economics analysis that is not very often used. I mean, not to say that we shouldn't do research, but often you do like the cool paper, and then that you don't actually see where it would go in the clinic.
So just to, first of all, I really agree with that, but just to play the role of like the optimistic or positive person, let's say, just for the sake of the discussion. I think like we're at the stage now that we're still very much limited only to like exploratory analysis and that, right? So does it make sense to do multimodal, right? Can it replace something? Can it offer something new? So we're not at the stage, of course, of like translating this in the clinic already, because as you very well said, it's very premature. We don't know if it will be better, if it would definitely it won't be more, it will be more costly and so on. So I think along these lines, what is very, very important, we discussed already about it, is the patient cohort, right? You want to have a big patient cohort that, not only big, but you want your cohort also to represent the variability of the disease, right? So how you select your patients is very, very important. But, it also, we could think of it as, at least for the future, maybe, for example, by, combining modalities, or at least my hope is not that in the future, the patient that comes in the clinic would need to undergo all these different tests. But maybe, for example, we find a biomarker by, like multiplexed imaging, or, single cell RNA-seq that somehow you can detect in a liquid biopsy, for example. Maybe you can predict something in a much, less cost-effective way. Or you then, you find it by combining data, it points you to something, and then in this way, when you actually translate that in the clinic, it would make some sense. But of course, to do that, we need really big studies, we need clinical follow-up, right? It will take a lot of time, and we really need validation in a new cohort. So you need to discover it in one cohort and validate it in an independent cohort. Because, of course, when we talk about translation in the clinic, it's not just about publishing cool papers, right? So, but I think there is promise in this area, I would say.
And on the question of modalities that have to add something in the health economics manner, there are also current modalities that are very expensive. So, for example, you can think of genetic tests in cancer that give access to some drug, some targeted drug, where we actually can predict quite well the expression of that gene from histopathology slide. And so that's much cheaper actually than doing the genetic test. It's maybe 30 euros to do the model inference on the slide, and that way you can get a pre-screening, so it's obviously less precise than doing the actual genetic test, but you can get a pre-screening, and that then goes to the genetic tests. And when there is a low prevalence of those genetic mutations in a population, that can really make it worthwhile to really look for that mutation, to have a cheap pre-screening test. And we do that for MSI. We are developing it. Yeah.
Thank you. So I'm thinking about, it's very nice to have both like maybe realistic point of views, but also futuristic point of views in combination of together. But again, to your point, Jonas, about that, maybe now combining all the modalities may not improve that much the result of the diagnosis. But maybe we could think about it that we might, for example, in the examples of neurodegenerative diseases that you were talking about, maybe in different stages of the disease, we need just a limited number of combination of these modalities. So do you have some more optimistic opinion? Or you can say the realistic, of course.
But that's true. That's actually true also what you said. So this is exactly the case. So you have this, like in, let's say, liver cancer, right? You have this scoring system called LIRADS, and you have grades. You can just look at the image and then you do the grade. If you have a certain grade, I think it's 4 or above, then you say it's certain that this person has liver cancer and I don't need to do the biopsy, right? So this is built in, like in many of these tests. You have this successive thing. If you have sufficient evidence from one modality, you don't go and look for the other modality. So this is already the way, especially I think oncology is probably the best field for this, because you have these tumor boards and you have people that are used to actually sequencing the tumor, and you have all these fantastic things that are happening with the immunotherapy, like incredible, really incredible research. And here, this is really where you do a lot of effort, and based on that, you will modulate your therapy. And that's exactly what you said. So I think, yes, indeed, that depends on the stage. And it's the same, it's exactly the same for Alzheimer's disease, indeed. Like, you have, at some point, you have some threshold concentration where if it's above, you know, it's not so informative anymore. But if you're at the lower level, maybe it's informative. Also, just to the pessimistic versus realistic versus optimistic. Just to say, of course, I'm also an optimist, because we also do research. So we have a project on heart failure, which is exactly about these integrative things, right? That we will do, like transcriptomics and genotype and a bunch of stuff, plus the cardiac imaging. So we will do that, but this is a case, this is maybe like an interesting case. So this is a disease called heart failure with preserved ejection fraction. And it's people that pump enough blood, that they have enough like blood going through the heart. But these people, we don't know how to treat them. It's very heterogeneous. So the assumption behind the project was, okay, we need to find subtype, like the subgroup we discussed before. And if we find these subgroups, maybe we can find druggable targets to help these people, because currently nothing works with this. There's been so many trials and nothing works. So the idea is really to go back and do integrative approach. So I'm optimistic, but cautiously, because as you said, and this is an extremely important point, like the cohort size is the key. So what we're doing is we were discussing at coffee before, but we're starting with a UK Biobank dataset, which is, for those of you that don't know, it's an incredible dataset. This is, I think, one of the greatest success of modern science, UK Biobank dataset. Check it out if you haven't. The other one being Allen Institute for Brain Science. These two projects have basically changed the whole field of everything. It's incredible. So UK Biobank is 500,000 patient subjects plus that have genotype and all the clinical data you want. It's a community sample in the UK, as the name suggests. Out of these, 100,000 will have imaging, and currently they have like 42,000, which have heart imaging and brain imaging, and also I think eye fundus imaging, maybe for already close to 100,000 for these people. So this is incredible resource for imaging genetics. They also have all the genotypes. So this means for us, we can actually build our discovery cohort on this and then validate on the patients. We can, because in Switzerland, how can you recruit? You don't even have that many people, right? It's like if you're talking Geneva and Lausanne, right? So this is that's, I think, essential what you said. Like this validation between two, that's the key. Like it's and that's more and more possible with the sharing platforms. In imaging, now you have Open Neuro. That's like basically you can publish your data sets. They're hosted for free. It's incredible. Like there's a lot of open data out there that really helps drive the quality up, I think.
I have actually a follow-up to your points, and I'm not sure how it's in academia, but in pharma at least, we wish we wouldn't have to do multimodal, but we have to, because a lot of the information, or at least for observational studies, are in disparate data sources, and our view on the matter is that potentially. This is due to that physicians might not have the opportunity to actually report a comprehensive view of the patient. So in your opinion, and we talked about this previously, the importance of data interoperability. Does in your point of view, hospitals have a place to play here to create a foundation where we can actually explore modalities or different modalities? Are physicians prohibited through their clinical information systems to actually measure multiple modalities?
Okay, so to this point, I think this is really a bit specialty dependent. I think, as I was saying, I think in oncology that's built in the culture now, that they have this thing. So you have people actually in Lausanne, like Michel Coindet, Olivier Michelien, that are doing like really cool stuff with like nice dashboards, visualization, integrating all these data sources. We also have an amazing data science team, by the way, at the hospital in Lausanne. They're really pushing a lot the envelope, trying to make everything interoperable. We have semantic graphs. We have a bunch of things. I'm really super happy with the IT at CHUV. It's really like they're really doing kind of pushing the envelope. So I think that this is really specialty dependent, this way of acquiring the data. Now, in terms of like as a data scientist or machine learning person, we now have access to most of the things. Now we're doing some work in a radiology department to put also the radiology metadata in there, so that you could search, people that have this type of imaging of this part of the body, with this diagnosis and this type of biomarker. So this is coming like in 6 months. We have right now the radiology is not there, but we're really pushing towards this so that you have this integrated view. We're also pushing a lot of between the hospitals to have this kind of standardized way, at least for the imaging data. We have the same platforms, we have the same way of it's stupid, but just organizing the files. How do you organize? What is the subject? That takes so much time. But now we have this standard called BIDS that developed by a lot of people, and that's what we're adopting, at least Geneva, Lausanne, and a few other hospitals. There's also the nice Swiss Personalized Health Network project, which tries to, at least make just one thing: depersonalization of the data. How do you depersonalize this data, this imaging? You would think just remove the age and stuff. No, it's actually it's just horrible. But now you have kind of a standard that agrees at least all the hospitals in Switzerland. So we're really in a place where people have realized the value of data. Also, the Swiss National Science Foundation have done an amazing job. By now, you have to open your data, and you have to open your publications and your code. So it's great. Like, you can start a lab with zero data. You can just grab a bunch of things and make some discoveries. So yeah, I think it's progressing.
Very promising. Thank you. Yeah, so unless there are any other questions to multimodality, I would say let's reserve some time for another very exciting topic. There is this field of causal machine learning, and up to recently, the field of machine learning and causal inference have developed independently, but now the two fields have found an interest in each other. So there's a huge potential for causal inference to improve generalizability of machine learning algorithms beyond distributions that are independently and identically distributed. And for causal inference, there's huge promise of using machine learning techniques to generate causal structures from data, as well as using methods for representation. So Félix, maybe to you, do you think that machine learning needs causal inference, or vice versa? Do you think causal inference can profit from machine learning?
Thank you. So yeah, I think the two are quite closely linked, and I think there's a difference we can make between two very distinct scenarios, especially when it's the AI and pharma track. So it's really, do you have a drug, or are you looking for a drug? So I think once we have a drug, the main causal question is to understand the treatment effect, the safety signals as well. And obviously for that we have the randomized controlled trials, and that's great and very easy on a causal way, because thanks to randomization, you know that the difference you observe is due to the treatment. But it's still limited, and I think there's been a very great example of the limitation of the randomized controlled trial with the Pfizer-BioNTech vaccine, and what they did with in Israel. And so in Israel, they basically Pfizer was able to access data on basically every Israeli patient, and that allowed them to have absolutely huge cohorts, observational cohorts, but still well characterized in order to have a very good estimation of the treatment effect. And they did a really amazing job. And so, for instance, if you compare the Pfizer-BioNTech vaccine trial, what the emergency use authorization was based on was only symptomatic COVID, and the proof of that the vaccine was reducing mortality from COVID-19, that only came from the Israel work, because it's such a rare event that you can't, even with 20,000 patients in the clinical trial, it wasn't enough events to demonstrate that the COVID-19 vaccine was protecting from COVID-19 death. And so I think what that illustrates also is that the main problem to do that is organizational, and Israel has really done a fantastic job of having great organization in order to be able to do those kind of studies at scale. But ML can also contribute, and we've done a paper at Okin on that question of the kind of methodology you can do. And so basically it's the same, an observational study. It's basically the same thing as an external control arm, and we were interested in that and looked into that. And what we saw was that ML can really add some statistical power to those approaches. Propensity score matching is not really optimal, and it's actually the message is a bit similar to my presentation on covariate adjustment. Basically, if you do outcome models that can predict some of the variation in the outcome in those observational studies, then you can increase statistical power while controlling your type 1 error. And yeah, so and then obviously there's once you have a drug, and then what if you don't have a drug? And then it's much more complex, many possible different genes that you want to consider as potential targets, and you want to understand that whether acting on that gene is going to act and help with the disease. And so here the causality looks more like a graph where you have all those, where you have a causal graph and between the genes, for example, in oncology, and you want to know whether if you act at some point in the causal graph, you can impact the disease progression. So it's a much more difficult problem, but obviously the causal questions are key to really find better targets that are going to be able to progress through the different clinical development phases. One great example of is Mendelian randomization, and it's often based on the UK Biobank Jonas mentioned, where you can validate some intermediary phenotype as a valid target. So there's a great example with the good cholesterol, so high density lipoprotein HDL, which was seen in observational studies to be really very predictive of good cardiovascular outcomes. So the Pharma developed some drugs to increase HDL, but they did not improve cardiovascular outcomes. And through Mendelian randomization, so basically predicting from the genome which people are going to have a higher HDL, you can actually do an instrumental variable analysis. And this showed that actually the link between HDL and good cardiovascular outcomes is not a causal link. It's just a correlation. We don't know what is the actual, what drives that correlation. And maybe it was too long.
Just a follow-up question. Do you think these advancements in causal inference can help observational studies become a first-class citizen? So currently it's still considered a second-class one. Do you think that would accelerate the adoption of external control for arms, for instance, that you mentioned, or use of observational studies for accelerated drug approvals?
So I think there was a lot of excitement on that topic, linked also to the 21st Century Cures Act that mandated that the FDA consider real-world data in addition to in the regulatory submissions. I think it's unrealistic to expect the randomized control trial to disappear and be replaced by external control arms. There are cases where it is already the case that historical controls can be used, but it's very specific case, rare diseases where basically you don't have any more patients once you've done your different phases, because you've included all the patients that have the disease in the clinical trials. So if you do a new clinical trial, you won't be able to recruit any patients. And but I think we have to think about all the other things that real-world evidence can bring, and so it won't replace phase 3 data, but it can really doing real-world evidence at scale, like Israel does, or even the UK Biobank. I think it really allows us to understand a lot of things better in how the treatment works, and also to be able to do better and faster pharmacovigilance, and be able to find earlier if there is a problem.
So maybe I can ask if there is any question among audience for our panelists.
Thanks for this great discussion. I was also reflecting on long COVID as Jonas, I think you mentioned about this radiology versus clinical disconnect. I don't remember. Paradox, yes. And all of these really complex diseases where multimodality or multimodal data can support, and how that actually links to the global burden of disease and also to health economics, and how to impact for perhaps prevention-related topics rather than just treatment, right? So diagnose earlier, prevent diseases earlier. So I wonder if you have any comments about how multimodal data can also have impact on those complex diseases and the general global burden of disease, to any of the panelists. Thanks.
So I'm definitely not an expert in MS, but something I read very recently, I was really like, I would say surprised to read, is, for example, that, and correct me if I'm wrong, but like having had the Epstein-Barr virus, for example, predisposes you to a great extent for MS. And I'm sure you can tell us exactly the details, and I'm not, okay. But it was like a very, very striking paper, right? That briefly said that, okay, there is a causal link between exposure to the Epstein-Barr virus, which is, I think, like 80% of the population has been through or something. Don't quote me on the numbers, really, not sure about that, right? And developing MS. So this would be like a great example how a very like different source of information might actually complement what we already understand about the patient, right? And ideally, of course, like the dream of everyone would be that we have like a holistic representation of the patient, including not only genetics, but also a number of environmental factors like microbiome, for example. We've seen it's linked to a number of different things, from how patients respond to treatment to their overall immune health and so on. So ideally, we would want to have like a holistic representation of the patient, and given that representation to be able to make more informed decisions. But I would like put it a little bit into context now, especially after 2 years of, a pandemic, that we already, in multiple countries, there were issues convincing the population to do a proven life, saving vaccine, right? So I think we're a little bit far away from like a utopia of, that if you're able to have like detailed testing about this and that, you would be able to optimize your health in the long run, not just survive, but also thrive. Because it seems like sometimes when we try to translate these things in society, it's not just what is in the papers and what is in, the research, but it's also how to convince people that certain things might be, of benefit. So I think what you say is, of course, like really important and reducing the burden of chronic disease. But there are some very basic steps we need to do for reducing that, like, you know, convincing people to stop smoking, eat a more healthy diet, and so on. And somehow I feel like society is kind of, I mean, I know we get into like not so scientific path now, but it seems like, you know, like when I speak to my circle, which includes a lot of researchers, for example, you know, based on my job and so on, we are all like into what is like the next best thing we can do for this and that to increase, I don't know, like 2% something. But then, you know, when you translate these things in society, sometimes it's very disconnected. And I think like the pandemic was a great example of that. So my opinion on what you say is that it would be a dream to be able to do that. But now we'll become the pessimist. And I will say that I think we are far away from that.
Maybe one comment, because this Epstein-Barr verse is very interesting. So there's another example that I found like amazing for like in favor of this integrative analysis. This is more like really exploratory, as you said, right, initially. But then sometimes you have insights that are just mind-blowing, I think. So there's this thing about Parkinson's disease, that there's a theory that it originates in the gut somehow, and then it propagates to the central nervous system, right? So there's so please correct me if I'm wrong, because I'm not a medical doctor, right? But there's apparently the vagus nerve connects the gut to the brain, and it's some people, you could have a procedure that you can cut this vagal nerve. And there was this study that found that people that have had this vagal nerve cut are like 90% less likely to develop Parkinson's disease. And so this was mentioned by Alain Daguerre at McGill, who's a big proponent of the network hypothesis of like brain disease, that says that the disease in the brain propagates along the networks of the brain. And so you could really have this theory that you could see this misfolded protein. It's also a proteinopathy, right? Parkinson's disease. So you have also this misfolded protein that propagate along the connections in the brain. And if you cut the source, then you cut the disease. But this, you can only find this if you have, microbiome, if you have like all this data and you happen to be looking at, I don't remember exactly the original study, but this is an example where this kind of multimodal analysis or integrative analysis maybe can lead to incredible insights. I mean, it's not the case, like I don't think these diseases are monofactorial. Many diseases, like especially neuropsychiatric disease, they're very different. They're like, you have hundreds of genes. It's very difficult to pinpoint them. But sometimes you have things like this that are just, and like the Epstein-Barr as well. It's incredible. So I think definitely it's worth, continuing just to understand the disease. I mean, doesn't mean we have a treatment, but at least we understand better, and that's worth it in itself. That's for sure. Yeah.
Are there any other questions in the audience? Sure.
So maybe a point on the global burden of disease. I think in pharma we want to find drugs that basically cure the problems once they're already there. And but yeah, I mean it's clear that it's better if we can prevent things. I don't know that machine learning has a big role in that. I think there are like structural problems in society that are often causing those diseases, and so obviously there are like the social determinants of health and disease. So poor people have worse health outcomes. But there's also like even our transportation system is a large cause of our diseases. If people drive instead of walking or cycling, that's a whole lot of chronic disease that come from simply having a transport system that favors cars and makes the sedentary lifestyle easier. So yeah, I don't know that machine learning can help a lot, but
so with that, I think I would conclude with some key messages. For one, it seems there is still a lot of work to be done in multimodal learning. So we've heard some promising cases, but we're still figuring out how to find optimal representations. Likewise, there seems to be a lot of promise in causal machine learning. while it will never replace RCTs, it might increase adoption of observational studies. Yeah, I'm not sure if you have anything to add, Naghmeh, besides these 2
That
points.
was it all, and yeah, I think again it was good to see both opinion, like more critical, but also futuristic opinion. That of course, especially like for example with the examples of neurodegenerative disease, having like working more and bringing the understanding of these new modalities of data could help us even when we don't have the treatment, at least to earlier diagnosis of the diseases. Yeah, and thanks a lot, everyone, for sharing your experience and your opinion. And thank you all for staying with us till the end of the session. And I think there is an apéro now, yes?